Today our lecture is about biological degradation of pesticides. The contents: Introduction to biological degradation of pesticides, types of pesticides, pesticide degradation pathways, pesticide residues in food and the environment, pesticide regulations and policies, case studies of pesticide biodegradation, future directions in pesticide biodegradation research. Conclusion: Pesticides are widely used in agriculture and public health to control pests and diseases. However, their persistence in the environment and potential health hazards have raised concerns about their impact on human health and the ecosystem. Biodegradation is the breakdown of pesticides into less harmful compounds by microorganisms, plants, or fungi, is a promising strategy for reducing the environmental impact of pesticides. This presentation will provide an overview of pesticide degradation pathways, bioremediation strategies for pesticide-contaminated soils and water, and regulations and policies related to pesticide use and disposal. There are several types of pesticides used in agriculture, including insecticides, herbicides, fungicides, and rodenticides. Insecticides target insects and other arthropods that damage crops or spread disease. Herbicides target unwanted plants, while fungicides target fungi that cause plant diseases. Rodenticides target rodents that damage crops or spread disease. Pesticides can be classified based on their chemical structure, mode of action, or target organism. Common chemical classes of pesticides include organochlorines, organophosphates, carbamates, and pyrethroids. Organochlorines, such as DDT, have been banned in many countries due to their persistence in the environment and toxicity to non-target organisms. Organophosphates and carbamates target the nervous system of insects and can also be harmful to humans and other animals. Pyrethroids are synthetic versions of natural insecticides found in chrysanthemums and are often used in household insect sprays. Pesticides can be degraded through various pathways, including microbial degradation of pesticides, fungal degradation of pesticides, bioremediation of pesticides, phytoremediation of pesticides. Pesticides are chemicals used to control pests that can harm crops, animals, and humans. Pesticides can persist in the environment, leading to contamination of soils and water. Microbial degradation of pesticides is a process in which microorganisms break down pesticides into less harmful compounds. Types of microorganisms involved in pesticide degradation: bacteria, fungi, and actinomycetes are the main groups of microorganisms involved in pesticide degradation. These microorganisms have the ability to produce enzymes that can degrade pesticides into less toxic compounds. The effectiveness of microbial degradation depends on the type of pesticide and the environmental conditions. Microbial degradation of pesticides is a complex process that involves a wide range of enzymes and metabolic pathways. The genetic basis of microbial degradation has been extensively studied in recent years, and several key genes and gene clusters have been identified that are involved in pesticide degradation. The genes responsible for pesticide degradation are often located on plasmids or transposons, which can be transferred between bacterial strains and even between different species of bacteria. Many of the genes involved in pesticide degradation are inducible, meaning that they are only expressed when the bacteria are exposed to the pesticide. Some bacteria have evolved multiple pathways for pesticide degradation, allowing them to break down a wide range of different pesticides. Understanding the genetic basis of microbial degradation is essential for developing effective bioremediation strategies for pesticide-contaminated soils and water. By identifying the key genes and metabolic pathways involved in pesticide degradation, scientists can engineer bacteria that are more efficient at breaking down specific pesticides, or develop new bioreactors and phytoremediation strategies that are tailored to different types of pesticides. Microbial degradation of pesticides is largely driven by enzymes produced by microorganisms that are capable of breaking down the chemical bonds in these compounds. There are several different types of enzymes involved in pesticide degradation, including hydrolyses are enzymes that catalyze the hydrolysis of chemical bonds in pesticides, breaking them down into smaller, less toxic compounds. These enzymes are produced by a wide range of microorganisms and are involved in the degradation of many different types of pesticides, including organophosphates, carbamates, and pyrethroids. Oxidoreductases are enzymes that catalyze the transfer of electrons from one molecule to another, often resulting in the breakdown of chemical bonds. These enzymes are involved in the degradation of many different types of pesticides, including organochlorines, organophosphates, and pyrethroids. Lacases are enzymes that catalyze the oxidation of phenolic compounds, often resulting in the breakdown of chemical bonds in pesticides. These enzymes are produced by a wide range of microorganisms and are involved in the degradation of many different types of pesticides, including polycyclic aromatic hydrocarbons, PAHs, phenylureas, and anilines. Microbial degradation of pesticides is influenced by various environmental factors such as temperature, pH, moisture, and nutrient availability. Optimal conditions for microbial growth and activity are required for efficient pesticide degradation. Optimal temperature range for microbial degradation varies depending on the type of microorganisms involved in the pesticide being degraded. Generally, higher temperatures increase the rate of degradation, but extreme temperatures can be detrimental to microbial activity. The optimal pH range for microbial degradation also varies depending on the microorganisms and pesticides involved. Most microorganisms prefer a neutral pH, but some can tolerate acidic or alkaline conditions. Adequate moisture is necessary for microbial growth and activity. However, excessive moisture can lead to waterlogging and anaerobic conditions, which can inhibit microbial activity. Pesticide degradation requires a source of carbon, nitrogen, and other nutrients. The availability of these nutrients can limit microbial activity and affect the rate of degradation. Fungi are important organisms in the degradation of pesticides in soil and water. They can degrade a wide range of pesticides. Genetic basis of fungal degradation is the ability of fungi to degrade pesticides is often encoded by genes located on plasmids or transposons. These genetic elements can be transferred between different fungal species, allowing for the spread of pesticide degradation capabilities. Pesticides, including organochlorines, organophosphates, carbamates, and pyrethroids. Genetic diversity in fungal degradation. Fungal species exhibit a high degree of genetic diversity, which can affect their ability to degrade pesticides. Some fungal species have evolved to be highly efficient in degrading specific pesticides, while others may not be able to degrade them at all. Understanding the genetic basis of fungal degradation can help in the selection of appropriate fungal strains for bioremediation purposes. Environmental factors, such as temperature, pH, moisture content, and nutrient availability can all affect fungal degradation of pesticides. Pesticide properties, the chemical structure, solubility, and volatility of the pesticide can affect its susceptibility to fungal degradation. Fungal species and strain. Different fungal species and strains have varying abilities to degrade pesticides, and some are more effective than others depending on the specific pesticide involved. Co-contaminants. The presence of other contaminants in the soil or water can affect fungal degradation of pesticides by competing for resources or inhibiting fungal growth. Fungi produce a variety of enzymes that can degrade pesticides. These enzymes are typically extracellular and secreted into the surrounding environment. Some of the most important enzymes involved in fungal degradation of pesticides include lacases. These enzymes are involved in the breakdown of phenolic compounds, which are commonly found in herbicides and fungicides. Lacases have also been shown to degrade polycyclic aromatic hydrocarbons PAHs, which are common contaminants in soil and water. Peroxidases. These enzymes are involved in the breakdown of a wide range of organic compounds, including pesticides. They are particularly effective at breaking down compounds that contain oxygen or sulfur atoms. Esterases. These enzymes are involved in the breakdown of esters, which are common in many pesticides. Esterases are also important for the degradation of other organic compounds, such as fatty acids and triglycerides. Ligninolytic enzymes. Fungi produce a variety of ligninolytic enzymes, including lignin peroxidase, manganese peroxidase, and lacase, which are involved in the degradation of lignin and other complex organic compounds. Cellulolytic enzymes. Fungi also produce cellulolytic enzymes, such as endoglucanases, hexaglucanases, and chelobiohydrolyses, which break down cellulose into glucose and other simple sugars. Xenobiotic metabolizing enzymes. Fungi also produce xenobiotic metabolizing enzymes, such as cytochrome P450 monooxygenases and glutathione S-transferases, which are involved in the detoxification of pesticides and other environmental pollutants. Pesticide-contaminated soil or pesticide-contaminated water can be treated using bioremediation techniques that involve the use of microorganisms to degrade the pesticides into less harmful compounds. Bioreactors for pesticide degradation. Bioreactors are commonly used for the treatment of pesticide-contaminated soils. These systems involve the use of microorganisms to break down the pesticides in the soil. Bioreactors can be designed to optimize the conditions for microbial growth and pesticide degradation, such as temperature, pH, and oxygen levels. Phytoremediation of pesticide-contaminated soils. Phytoremediation is another technique that can be used to remediate pesticide-contaminated soils. This process involves the use of plants to absorb and break down the pesticides in the soil. The plants can then be harvested and disposed of, or the pesticides can be extracted from the plants. Phytoremediation can be an effective and low-cost method for remediating pesticide-contaminated soils. Plant microbe interactions in phytoremediation. Plant microbe interactions play an important role in phytoremediation of pesticide-contaminated soils. Certain plant species can form symbiotic relationships with soil microorganisms, such as bacteria and fungi, that can help to break down the pesticides in the soil. These plant microbe interactions can enhance the effectiveness of phytoremediation for pesticide-contaminated soils. Phytoremediation strategies for pesticide degradation. There are several phytoremediation strategies that can be used for pesticide degradation in soils. These include phytoextraction, where plants take up the pesticides from the soil and store them in their tissues for removal. Rhizodegradation, where plants release compounds from their roots that stimulate the growth of pesticide-degrading microorganisms in the soil. And phytostabilization, where plants immobilize the pesticides in the soil, preventing them from leaching into groundwater or being taken up by other organisms. Bioreactors are used for the treatment of pesticide-contaminated water and soil. Different types of bioreactors are used depending on the type and concentration of the pesticide, the physical and chemical properties of the contaminated medium, and the desired treatment efficiency. Common types of bioreactors include aerobic bioreactors. These bioreactors promote the growth of aerobic microorganisms that require oxygen to degrade pesticides. They are commonly used for the treatment of water contaminated with pesticides by promoting the growth of anaerobic microorganisms that can degrade pesticides in the absence of oxygen. Fixed film bioreactors. These bioreactors use a stationary medium, such as sand or plastic, to support the growth of microorganisms that can degrade pesticides. They are commonly used for the treatment of water contaminated with pesticides. Fluidized bed bioreactors. These bioreactors use a fluidized bed of sand or other small particles to support the growth of microorganisms that can degrade pesticides. They are commonly used for the treatment of water contaminated with pesticides. Suspended growth bioreactors involve the use of a suspended microbial culture that degrades the pesticides in the contaminated water or soil. Hybrid bioreactors combine the advantages of fixed film and suspended growth bioreactors. They use a solid support matrix to which microorganisms can attach and form a biofilm, but also maintain a suspended microbial culture to degrade the pesticides. The performance of bioreactors for pesticide degradation is affected by several factors, including pesticide type and concentration. Different pesticides have different chemical structures and properties that affect their degradation rates. The concentration of the pesticide in the contaminated medium also affects the degradation rate. Microbial population. The type and abundance of microorganisms in the bioreactor affect the degradation rate. Some microorganisms are more efficient at degrading certain pesticides than others. Nutrient availability. Microorganisms require nutrients, such as carbon, nitrogen, and phosphorus, to grow and degrade pesticides. The availability of these nutrients affects the degradation rate. Phytoremediation is a process that uses plants to remove pollutants from soil, water, and air. In the case of pesticide-contaminated water, phytoremediation involves the use of plants to absorb and break down pesticides in the water. Phytoremediation works by using plants to absorb pesticides from contaminated water. Once the pesticides are absorbed, the plants break down the pesticides through a process called phytodegradation. In some cases, the plants may also store the pesticides in their tissues, a process called phytoextraction. There are several phytoremediation designs that can be used for pesticide degradation in water. Some of these strategies include constructed wetlands. These are engineered systems that use wetland plants to treat contaminated water. The plants absorb and break down the pesticides, while the wetland soil filters out any remaining contaminants. Floating treatment wetlands. These are floating islands of plants that are placed in contaminated water. The plants absorb the pesticides and break them down, while the roots provide habitat for microorganisms that further degrade the pesticides. Aquatic plants. Some aquatic plants, such as water hyacinth and duckweed, can absorb large amounts of pesticides from water. These plants can then be harvested and disposed of, or the pesticides can be extracted from the plants for reuse or disposal. Phytostimulation. This strategy involves the use of plants to stimulate the growth and activity of indigenous microbial populations in the soil. This can be achieved through the addition of nutrients or other growth-promoting substances to the soil. Phytoextraction. This strategy involves the use of plants to absorb pesticides from the soil and accumulate them in their tissues. The plants can then be harvested and disposed of, effectively removing the pesticides from the soil. Phytodegradation. This strategy involves the use of plants to degrade pesticides through various mechanisms such as enzymatic breakdown or metabolism. This can be achieved through the selection of specific plant species that are known to have pesticide-degrading capabilities. Rhizodegradation is the process by which plant roots release organic compounds that stimulate the growth and activity of microorganisms in the rhizosphere. This can enhance the degradation of pesticides by these microorganisms. Phytoaccumulation. This strategy involves the uptake and accumulation of contaminants in plant tissues, which are then harvested and disposed of. This method is particularly effective for organic contaminants like pesticides, but may require multiple harvests over several growing seasons to achieve significant reductions in contamination. Plant microbe interactions is a process that uses plants to remove pollutants from soil or water. Plants can be used to absorb and break down the pesticides through a mechanism of interaction between plants and microbes in the soil, which can release compounds into the soil that stimulate the growth of microorganisms capable of degrading pesticides. These compounds can include sugars, amino acids, and organic acids. In turn, the microbes can help the plants by providing nutrients and protecting them from pathogens and other stressors. Mycorrhizal associations are symbiotic relationships between plants and fungi that can enhance plant uptake of nutrients and water. These associations can also facilitate plant uptake of contaminants such as pesticides, which can then be degraded by the plant or associated microorganisms. Pesticides can leave residues on crops that can be harmful to human health if ingested. These residues can accumulate in the body over time and cause adverse health effects. Regulations and policies are in place to ensure that pesticide residues in food are within safe limits. Pesticides can also contaminate the environment, including soil and water. This can have negative impacts on ecosystems and wildlife. Bioremediation strategies, such as microbial and phytoremediation, can be used to clean up pesticide-contaminated environments. Pesticides are widely used in agriculture to control pests and increase crop yield. However, they can also have negative impacts on human health and the environment. As a result, there are regulations and policies in place to ensure safe and responsible use of pesticides. In the United States, the Environmental Protection Agency (EPA) is responsible for regulating pesticides under the Federal Insecticide, Fungicide, and Rodenticide Act (FIFRA), which requires that all pesticides be registered with the EPA before they can be sold or distributed. The EPA evaluates the potential risks of pesticides to human health and the environment before granting registration. The EPA also sets limits on the amount of pesticide residues that can be present in food and drinking water. These limits are known as tolerances and are based on the EPA's risk assessments. The Food and Drug Administration (FDA) and the United States Department of Agriculture (USDA) are responsible for enforcing these tolerances and ensuring that food and water are safe for consumption. Other countries have their own regulatory agencies and policies for pesticides. For example, the European Union has a comprehensive regulatory system for pesticides under the Plant Protection Products Regulation (PPPR). The PPPR requires that all pesticides be authorized before they can be sold or used in the EU. The authorization process includes an evaluation of the potential risks to human health and the environment. Overall, pesticide regulations and policies aim to ensure that pesticides are used safely and responsibly to protect human health and the environment. Several case studies have been conducted to investigate the efficacy of bioremediation strategies for pesticide-contaminated soils and water. Here are a few examples. A study conducted in Iowa found that the use of bioaugmentation, a process that involves adding microbial cultures to contaminated soil, significantly increased the rate of atrazine degradation. The researchers found that the addition of a bacterial culture called Arthrobacter eschens DC1 was particularly effective in degrading atrazine. A study conducted in India found that the use of vetiver grass was effective in removing lindane, a highly toxic pesticide, from contaminated soil. The researchers found that the roots of the vetiver plant were able to absorb the lindane, which was then broken down by microbial activity in the rhizosphere, the area of soil surrounding the roots. A study conducted in China found that the use of a microbial consortium, a mixture of different microbial cultures, was effective in degrading chloropyrifos, an organophosphate pesticide, in contaminated water. The researchers found that the microbial consortium was able to degrade up to 90% of the chlorpyrifos within 7 days. Chlorpyrifos is a commonly used insecticide that can contaminate water sources. A study conducted in India demonstrated the effectiveness of phytoremediation in removing chlorpyrifos from contaminated water. The researchers used water hyacinths, a plant that is known to absorb and accumulate pollutants from water. After a period of 6 weeks, the concentration of chlorpyrifos in the water decreased significantly. Lindane is a pesticide that can persist in soil for several years. A study conducted in Spain demonstrated the effectiveness of fungal degradation in removing lindane from contaminated soil. The researchers used a fungus called Trametes versicolor, which is known to produce enzymes that can degrade lindane. After a period of 6 months, the concentration of lindane in the soil decreased significantly. DDT is a persistent organic pollutant (POP). In a study conducted by Jabin 2018, a phytoremediation strategy was employed to remove DDT from contaminated soil. The researchers used a plant called vetiver grass, which is known for its ability to accumulate pollutants in its roots. The researchers found that the vetiver grass was able to remove up to 80% of the DDT from the contaminated soil after 90 days of growth. The researchers also observed that the DDT was broken down into less harmful compounds by microbial activity in the soil. As research on the biodegradation of pesticides continues, there are several promising areas of investigation that could lead to more effective and sustainable bioremediation strategies. As the negative impact of pesticides on the environment and human health becomes increasingly apparent, there is a growing need for effective and sustainable bioremediation strategies. Future research in this field will focus on several key areas: identification and characterization of new microbial and fungal strains with enhanced pesticide degradation capabilities, development of more efficient and cost-effective bioremediation technologies, such as genetically modified microorganisms and nanotechnology-based approaches, investigation of the potential of natural plant microbe interactions for pesticide biodegradation, and the development of new phytoremediation strategies, assessment of the long-term effectiveness and sustainability of bioremediation strategies, and their potential impact on the ecosystem. Metagenomic approaches involve analyzing the genetic material of entire microbial communities rather than individual organisms. This can provide a more comprehensive understanding of the microbial processes involved in pesticide degradation and identify previously unknown biodegradation pathways. Synthetic biology approaches involve engineering microorganisms to enhance their ability to degrade specific pesticides or to produce enzymes that can break down pesticides. These approaches have the potential to create highly efficient and targeted bioremediation strategies. Nanotechnology involves the use of nanoparticles to enhance the effectiveness of bioremediation strategies. For example, nanoparticles can be used to increase the surface area of microorganisms, allowing them to more efficiently degrade pesticides. Nanoparticles can also be used to deliver nutrients or other compounds to microorganisms, enhancing their growth and activity. Phage therapy. Phages are viruses that infect and kill specific bacteria. Phage therapy involves using phages to selectively target and eliminate pesticide-degrading bacteria that may be inhibiting the biodegradation process. This approach has the potential to enhance the effectiveness of bioremediation strategies by removing competition and allowing other microorganisms to thrive. Pesticide biodegradation research has made significant progress in recent years, but there is still much work to be done. As pesticide use continues to increase, the need for effective and sustainable methods of pesticide removal from the environment becomes more urgent. Future research should focus on developing new and more efficient methods of pesticide biodegradation, as well as identifying and characterizing the microorganisms that play a key role in this process. Additionally, there is a need for more research on the impact of pesticides on human health and the environment, and how biodegradation can help mitigate these effects. Overall, continued research and development in the field of pesticide biodegradation is crucial for ensuring a sustainable and healthy future for our planet. Thanks. You can find a link below in the description to download a free copy of the presentation. The link takes you directly to Google Drive. Please like, subscribe and write a comment. Please don't sell the presentation or upload the file to another websites, because it's copyrighted. So what is the next video? Write down in the comments section the topic of the presentation and the number of slides. See you in the next video. Presentations upon your request.